Hey everybody, it's Mrs. Mancuso with some more AP Bio review. So, cells. All right, we could go into all the organelles, but I feel like that's something that you really should know already. So I'm going to pass by those, and I'm going to go right to membranes. So membranes, every cell has an outer cell membrane. What's unique is eukaryotic cells have membranes inside, and a membrane is a phospholipid bilayer. So a phospholipid, has a polar head, which means it has a charge. It's negative, and that's due to the phosphate group that's on it. And then it has two fatty acid tails. The fatty acid tails are nonpolar. So because there are portions of the molecule that are polar and portions of the molecule that are nonpolar, it's amphipathic. And they line up like a sandwich like this, which is why it makes such a great selectively permeable bilayer. For something to get through, it has to be able to pass through the tails. So here's my little molecule. If it wants to enter, it has to be acceptable by the tails to make it through. So things that can diffuse right through, that's just simple diffusion. That's things like oxygen gas, because it is nonpolar. So things that are small and don't have a charge can go right on through the bilayer. Now, a lot of things can't pass by, so they have to go through channels. So here's a channel. It's a protein, and this channel often has a gate, and when the gate is open, things can enter. An ion, like sodium, is a great example of a molecule that can't get through the bilayer because the polarity, the charge of this molecule is not liked by the nonpolar tail, so it can't get through, even though it's small. So instead, it has to go through a gated ion channel. So here's my gated ion channel, and it lets sodium in. Sodium will flow from high to low. So this is called facilitated diffusion. Facilitated because it's using the help of a membrane protein. This membrane protein is also amphipathic. It must have regions here that get along fine with the tail, so this region of it's got to be nonpolar, and this exterior part of it has got to be polar. So both lipids and embedded proteins that are integral, that go all the way through, are going to be amphipathic. All right, there are sometimes molecules, and I can use sodium again, but they have to go through a pump. So if this is my pump, this can be a sodium-potassium pump, which we see in nerves. It can move sodium out from low to high. So if I move sodium against the gradient and I stockpile it here, that's going to require ATP. So any any membrane protein that requires ATP is a pump, and this is active transport. So the difference between facilitated diffusion and active transport is the need of ATP because you're going against a gradient. All right, we did some labs, so I really just want to go over the labs because I think that the AP really likes to see that you guys have done the labs. So we did 4 different labs involving membranes. The first one was just a diffusion lab. And in the diffusion lab, you had a container, like a plastic cup. And in the plastic cup, you put starch. Oh, no, sorry. No, you didn't. You just put water and iodine. And then you made one of those cells. You put starch inside the cell, and you put glucose inside the cell. And it was diffusion, so we were trying to see like what's moving in and what's moving out. And what we found out was that glucose can move out and iodine can move in. Whether or not water moved or not, we could have masked it before and after and figured that out. We just didn't in this lab. So the way that you test for glucose, the way you know if it moves or not, is you use Benedict's solution. And Benedict's solution starts off blue, and you heat it. And if there are sugars present, it will change green to yellow to orange to red based on how much sugar is present. A little bit of sugar, it'll turn green. A lot of sugar, it'll turn red. But you have to heat it up for like 5 minutes to have that work. So, of course, this tested positive inside for Benedict's solution because I put glucose in there. But this water after 24 hours also tested positive for glucose, proving that glucose moved out. The test for starch is iodine. So when starch and iodine are both present, you get this blue-black color. If it's just iodine, it looks an amber color. So out here at the beginning of the experiment, it was amber out here and it was clear inside. And what happened was after 24 hours, of course we get a positive starch test outside, or a positive, oh my gosh, we still have an amber color outside. So we didn't get a positive test for starch on the outside because starch is too big, so starch didn't move. But inside, we definitely got a positive test for starch because starch started out there. And the iodine moved in. So the iodine is small enough that it could diffuse in, and we got a positive test. All right, sorry, that was like a rough one. Easy lab, but rough with my talking right now. All right, osmosis. This is the movement of water. So we want to set up two different solutions, two different cells. One of them put sucrose in the bag and water outside. The other one put water in the bag and sucrose outside. Mass them first, and then leave them overnight, and then mass them again. And what happens is this one, the water is hypotonic, and the sucrose is hypertonic. Water always moves from a hypo to a hyper, so this one gained mass. I'm going to put a big plus because it gained mass. In this one, my water is hypo, this solution is hypertonic, so water is going to move out, so this one lost mass. So that was our osmosis. The next one that we did was called plasmolysis. And you grab the microscopes and you grab some, I think we had green onions and maybe some Eladia. And when you look at a plant cell under a microscope, you can definitely see the cell walls. They look very rectangular. You'll see a lot of cells next to each other like this. You can't see the membrane very well, but you see chloroplasts all the way to the edges. So that's how you know the membrane goes all the way to the edges. So you could see all this. And so that's just a normal plant cell. There's actually turgor pressure, water pushing on that membrane to keep it nice and stiff so that the plant doesn't shrink. Well, if you go putting this in salt water, what happens is the cell wall stays the same size. So this still looks the same as it ever did. But all of a sudden you see just a clump of the green in the middle. That's every bit of your membrane has shrunk in. So now your cell membrane is right here. So what happens is this was hypotonic and the salt water was hypertonic, so it lost water. And as it lost water, it shrank. And this will actually start to pull away from the wall, and that's what plasmolysis is. And this is what causes wilting, because these walls will collapse on each other and the plant will go and fall over. All right, the last lab that we did, our fourth one, was about water potential. And this is by far my favorite of the 4 labs. You took and you used a little cork borer and you cut out little potato skins. They had no skin on it and they were around all the same size, and you masked them first. Then you put them in all these different solutions. So we had distilled water, we had a 0.2, a 0.4, a 0.6, a 0.8, and even a 1.0. And you put your little potatoes in there overnight, and you see how they change in mass. And we make a graph like this. This is 0. This is percent change in mass. So if I have distilled water and I put potatoes in it, the potatoes are definitely hypertonic and the water's hypotonic. So it's going to gain mass. So there it is. And then we have 0.2, 0.4, 0.6, 0.8, and 1.0 here. And what you do is you figure out the percent change and you plot your points. And then you make a best fit line going through. Wherever it is at 0, wherever it crosses the axis, that is equal to isotonic, meaning it's not gaining or not losing. So that is equal to the value C. C is the concentration of sucrose inside the potatoes. I'm going to use that C in my water potential formula. So water potential is equal to solute potential plus pressure potential. This only happens for cells, and I need to know the turgor pressure for this value. But what I can solve is this. This is equal to negative ICRT. So negative is an ionization constant that's equal to 1.0 for sucrose. C, I go back to my graph and I say, all right, let's say that was a 0.3. R is a pressure constant, 0.0831. It's on your reference table. T is the temperature and it has to be in K. So let's say it was 300 K. Then I take my calculator, my Yankee calculator, and I do my math. 0.3 times 0.0831 times 300. And I get negative 7.5 bars. Really important if this is a math problem that you remember to put that negative on there. But that shows me what the water potential is. So if I have a cell in a solution, this is negative 7.5. That's my potato. Depending on what I put out here, if I put something that's negative 5, think of it like a thermometer, negative 5 is higher than negative 7.5. This is going to be my hypo. This is going to be my hyper, and water will move in. If instead my potato is negative 7.5 and I put it in a negative 10 is lower, so that's hyper, and now my potato is hypo, and out it goes. So some of them are going to gain mass and some of them are going to lose mass based on the solution. All right, thanks for joining me.